Hi everybody, thanks for coming for this GSI technology series. As you all know, genomics technologies are developing at a very high pace. For example, single cell matured, we started using it in 2017, and by now it is pretty mature technology that allows you to get pretty good quality data. But the main shortage of single cell transcriptomics is that you basically lose all the information about where these cells were in the tissue. So the spatial component is completely lost when you dissociate the tissue, right? So then spatial approaches appeared and they work reasonably well, but they're still, I wouldn't call this a mature technology already. There are still many things going on. And the major limitation is usually you doing not a whole transcriptome approach, but probe-based, limited to just a subset of the targets. Or if it is whole transcriptome approach, there are other limitations like sensitivity, but possible diffusion, etc. The approach that you will be hearing about today was developed by a company called Curio a while back, and it seems very promising, and a very established company like Takara eventually decided to purchase this Curio company. So that approach allows you basically to barcode the tissues with spatial barcodes and then dissociate the tissue and get the full advantage of single cell good quality data. It's compatible with the next genomics and probably some others. So it's my pleasure to introduce today's speakers, Ozgi Getkin from Takara and originally from Curio, right? And also Nick Vysotsky, who is account manager for Takara, who will later then give you some overview of other Takara solutions for just RNA, from low input RNA, etc. So please, guys.
All right. Thank you very much for that introduction, Yuri. Can everyone hear me just fine? I can't quite tell up here if I'm speaking too loud or just right. How we feeling? Thumbs up? Going all right? All right, I know you got your hands full on over there. But thank you everybody for coming on out today, taking time out of your busy schedules to get some food and listen to us share some insights into our technology, the spatial omics and RNA-seq solutions. Yuri already gave a wonderful introduction here, but we are your Boston University dedicated team. Picture myself and Ozgi there. We are local representatives. If you guys ever have any kind of questions, quote requests, requests for technical insight or assistance, we will be your primary contact to reach out to and help get you some answers or provide some insight. As far as our lunch and learn, a brief agenda here, an overview of Takara Bio USA, who we are, how we got our start, where we're at today, a high-level overview of our RNA-seq technologies such as SmartSeq mRNA-LP, our newly launched SmartSeq mRNA long read, before handing the mic on over to Ozgi to discuss some spatial omics, including checker mapping, secret transcriptomics, and then leaving some time at the end for open discussion and questions. So, Takara Bio, what do we do? Our core businesses, we develop innovative solutions for genomics, cell analysis, and molecular biology, by providing a wide variety of enzymes, kits, instrumentation, and services. Although this is what we do today, it's far from what we did in the beginning. We got our start in 1842 brewing sake in Kyoto, Japan, a little known fact, but I think particularly interesting to our story. After dipping our toe into the world of biology, after that we dipped our toe into the world of biology with yeast experimentation in the mid-1900s, and then established Takara Bio in 1967 as an offshoot of the Takara Sake brewing company. So that was about 60 years ago, so we've only been a biotech company for about 30% of our total existence. And just a quick little snapshot. We still do make the sake today, so we are two separate. I don't know if you guys saw the sake bottle sitting on the front on over there. Wonderful sake, highly recommended if you guys can find that in stores. It's great stuff. Continuing on with our history, after Takara Bio Incorporated was established, we acquired CloneTech in 2005. So a lot of you may be familiar. They had developed the SmartSeq technology as well as some of our most popular PCR enzymes, such as titanium TAC, our cloning and fusion cloning, our Lenti-X platform, if you guys are familiar with that. Before moving on in 2017, expanding further with the acquisition of Rubicon Genomics, providing the library prep that we incorporate into our kits now, especially for cfDNA and FFPE, some of those degraded samples that really assisted in expanding, us there. Relocated from Mountain View, California to San Jose in 2021. That's what the picture is of here, our shiny new headquarters. And we greatly expanded our space for R&D. So really setting ourselves up well for the future. And most recently acquiring Curio Biosciences with Ozge and Jane in the background over there. Developed locally within the Broad Institute, and that will constitute the majority of our presentation today. But with that, our history from sake all the way to expanding our biotech sphere of influence. We have a whole bunch of product offerings now, from humble beginnings of yeast experimentation all the way to PCR, next-gen sequencing, spatial genomics, viral delivery. We've really expanded in such a way that we touch just about every single area of molecular biology research. But today, we're going to be focusing specifically on next-gen sequencing, the mRNA-seq, as I explained a little bit earlier, and then spatial genomics. So starting with a very high-level overview of our product line, we provide RNA-seq for immune profiling, anyone who's looking to do TCR, BCR profiling, mRNA-seq for precious samples, high-RIN samples, single-cell, total RNA-seq for more of your degraded samples, maybe FFPE, as well as small RNA-seq. Now I know Yuri is pretty familiar with our total RNA-seq. He's incorporated that into the core in the past, but today I saw a lot of you guys were signing up saying you're most interested in single cell, working a lot with those fresh frozen samples. So we're going to be focusing on the SmartSeq mRNA single cell portion of our portfolio. So SmartSeq mRNA, now what is it? Now SMART stands for switching mechanism at the 5' end of the RNA template. Over to the right, I have a graphic. I hope some of you guys in the back are able to see that just fine. Hopefully it's not too small. But essentially how it works is you start with your mRNA, you oligo DT prime it, and perform your first strand synthesis, which is then has tailing by reverse transcriptase. So we have a SmartScribe RT, which is a modified NMLV reverse transcriptase, which adds a known non-template sequence onto the end of that first strand, which is the kind of 5 X's denoted by the 5 X's there with the 3' end. Now with that known sequence of the non-template, we were able to develop our SmartSeq mRNA template switching oligo, which is able to anneal to that known sequence. From there, the template switching mechanism occurs, and it extends to fully incorporate the primer into the original mRNA template strand. And then you're ready for PCR to be able to be amplifying and continuing on to your library prep. So with this workflow, you're able to create high quality, full length cDNA libraries from low input samples. Because it is full length, you're able to identify novel genes, isoforms, and splice variants with confidence. Basically, you're getting the full picture, so you're getting full results. You don't need to worry about having left out any information that could be crucial to your research. And all this is a single tube cDNA synthesis workflow. This process right here takes about 2 hours. So relatively quick, don't need to worry about any spillage or mixing up their samples in a single tube there. Any questions currently? All right. So continuing on here, diving in a little bit deeper into our SmartSeq mRNA. It's a very well trusted sequencing option. It's true full-length mRNA-seq with over 3,000 publications. I have a publication here to the right. It's more of a comparative analysis of our single cell sequencing options. It's compared to study 6 different RNA sequencing methods. Found that our SmartSeq was the most sensitive and accurate on the market, and it recommends it for your sequencing purposes. As far as input range for SmartSeq mRNA, it ranges from 10 picograms to 100 nanograms total, or 1 to 1,000 cells. It allows for direct input. This is the only commercial solution that allows you to do that direct single cell input. So it allows you to maximize your insights, not your sample loss. It's also ideal for precious hard-won samples such as organoids for disease modeling, regenerative medicine, etc. Now, if we look for even higher accuracy than this kit already offers you, there are UMI options available, and we do have analytical user-friendly tools through our Cogent software online. It's available for free download, where you can be performing your analysis after your sequencing. Now, continuing on from our SmartSeq mRNA and building upon the SmartSeq mRNA workflow, again, we have our newly launched SmartSeq mRNA long read, which, as you can see there, it utilizes essentially the same workflow as our SmartSeq. One of the only differences is that it incorporates SmartSeq long read template switching oligos compared to our regular SmartSeq oligos. And then for your library prep, it incorporates ONT, library prep, and sequencing, which ends up setting you up pretty nicely if you want to get into spatial beyond that. Now, again, working through the workflow, it's DT prime CD synthesis. The bar-coded cDNA is pooled for a single reaction library prep, multiplex up to 96 samples in a single tube. The workflow itself is about 1.5 days. And at the end of it, you're able to obtain high-quality sequencing data with average read lengths of about 2 kb and detect transcripts up to 8 kb. And it's compatible with Curio spatial. Just a quick little snapshot of some of the key performance data from their SmartSeq long read. The mRNA long read generates full-length cDNA with a wide read length distribution. cDNA size there, I don't know if that's too small for some of you guys in the back, but about an average size of 2,500 base pairs, whether you're going in 10 picogram inputs, single cell, or up to 10 nanograms. And the read length distribution is very similar, with 10 picograms about 1,200 NT, and 10 nanograms, 1,400 NT. So good size distribution, and read length distribution. So very quick high-level overview of our SmartSeq mRNA technologies and our newly launched SmartSeq mRNA, single cell and long read, my apologies. And that's good science. Want to save some good time for Ozge here in our spatial, but you guys got any kind of general questions about our SmartSeq technologies?
The last one, is it single cell per well?
Single cell per well. You know, I'd have to double check on that. I'll double check for you.
It says 100 samples at one plate. You put 1 cell per well.
My apologies. I need to double check on that for you. Anybody else? Curious questions? All right. Well, thank you for indulging me here and sharing some of our SmartSeq technologies. I'll make some room for Ozge here to continue on with our spatial technologies for you guys. Thank you very much.
Thank you. And we both will be here after our talks if you want to have individual talks, but we will also follow up with the slides and the emails. And if you have not, I guess it is not working. Is it better? Okay. And if you haven't registered and if you fill in your information email, we would really appreciate that so that we make sure that you get everything, and any question we can respond right away, we will be able to follow up. So thank you. And I'm going to try to use this. If I cannot manage, I will quit on this. Right? Is that? Just presentation here, right? Did you change anything? Perfect. So I am, hopefully it didn't bring me all the way to the beginning. So that's a quick review. And now here, I don't know why this does it. What did you do to my slide? Right.
It's just the other PowerPoint tab.
Yeah, I mean, from the presentation mode, it brings up. All right, perfect. I'm not going to use my tool. And so I'm Özge, and I am the spatial specialist, and I like to turn to sir. And I really appreciate spatial in more personal level, because I spent so many of my years at MGH core, neuroscience core, doing a lot of laser capture, and you can imagine how long that takes to capture 2,000, 5,000 neurons. So I have a very strong bias to spatial biology. This is, I don't know, is it your slide? You didn't add mine. I'm just gonna find my deck. I apologize. So I am safer here. Connected. Yes, that is me. So, thank you so much for joining today, and I'm really excited to show you spatially tracker. I'm gonna focus. I'll mention the seeker. But the tracker is basically taking your single cell sequencing data and convert that to a spatial map. So today I'm going to walk you through how we do that. But before that, let me give you a little overview of the single cell sequencing. In the last decade, single cell technologies have exploded. And higher throughput, more modalities, more resolution, we wanted more. Technologies like DropSeq and CytoSeq, pioneered by our Curio co-founders from Broad, opened the door to high throughput single cell sequencing, and SmartSeq is originally licensed by Clontech, now Takara Bio. So we have really masterminds on the single cell and the spatial. And the single cell sequencing gave us incredible insights into cellular diversity. We know now who these single cells are, and we are missing some big part of the question that we're all trying to ask, which is, where are these cells in the tissue? Yes, UMAP looks great, and we can cluster cells perfectly, but the cells in real life don't sit on clusters, and biology is fundamentally spatial. And the cell functions depend on where they are, who their neighbors are, and what they are responding to. And that is why spatial biology emerged, and why we are here today, and why it's so popular. Most current spatial methods today fall into two general categories that we are all familiar with. Either they are capture-based and sequencing readout platforms that heavily rely on the convolution to call out these transcripts in a cellular boundary, or you use probe-based, similar to in silico hybridization, microscopy readouts, where you need segmentation, relying on these images to call the cell boundaries. And while these spatial platforms have delivered real breakthroughs, understand the neuroscience and tumor microenvironments, they still come with substantial limitations and challenges, especially expensive instruments, heavy bioinformatics burden, and the convolution segmentation bias. And none of them really offer single cell resolution, and I will walk you through that in more detail. So spatial biology today reminds me NGS a decade ago, when the bioinformatics were impossible. When people first came and asked single cell experiment from LCM, I thought they were crazy, and it really required a lot of knowledge, a lot of resources and funds. And so we need spatial resources that lowers the barriers and that is more accessible. So let's look at the resolution issue. So current spatial technologies only approximate single cell resolution. What do I mean by that? So imaging-based, probe-based methodologies are great. They have really high resolution, and then you can get subcellular, but most likely you have to find the perfect segmentation markers, and not every antibody, not every key is gonna open the same doors, and the cellular differences in different tissue or complexity is gonna be challenging. And then you think about capture-based, somehow easier, but then you are going to be capturing transcripts and try to draw out cell lines through deconvolution. And the quality of your data depends on how well your single cell RNA annotation is, and the bias and the noises, everything will affect you. So either methodology sits somewhere on a resolution spectrum. One is too high, one is too low, but both are approximate interferes that single cell resolution. So this is where the new class of spatial biology comes in. And I try to put it in a different category, which is donation-based tagging technology. Tracker offers single-cell spatial mapping through a kit. Let's walk how we do that and what that means. We basically take your single-cell data set you're already generating, that you already mastered your bioinformatics with, that you're comfortable with your workflows, and by tagging each nuclei while still in tissue and intact, and we then convert them to spatial maps. So with a single workflow, you get single-cell transcriptomic resolution through single-cell architecture with no segmentation, no deconvolution, and you don't need any expensive instrument. So let's see how Tracker does that, starting with how we tag each nuclei in place. The foundation of Tracker is the slide tag technology right from the Broad Institute that Fei Chen and Evan McCaskill, also the co-founders of the Curio, and it based on a Takara slide with a 10 by 10 tile. And this tile, 10 by 10 millimeters, so 1 centimeter square, covered with a closely packed monolayer of beads, and each carrying oligos with spatial barcode. So these two slides, I'm going to slow down because this is where all the magic happens, and I will pause for questions if you have further questions. So this barcode have 3 distinct regions. One is this essential part photocleavable linker, so that barcode can be released upward to the tissue, as we talked about donation, and a spatial barcode which gives each nucleus spatial identity, and a handshake sequence which allows seamless compatibility with standard single-cell platform like Penix, BD Rhapsody, Parse, and we are Illumina. We are working towards being compatible with any downstream platform you are using. So together, this lets us attach spatial information directly to the nuclei and map them back to their exact location with true single-cell resolution. And most spatial technologies that we know, and this is where the diffusion seems like a concern, works on capturing the transcripts into a surface, captures a surface. And then you have to infer through the convolution where each cell came from, or what transcripts align with what cell content. And Tracker flips this strategy. Instead of capturing RNA, we donate spatial barcodes directly to the nuclei. So upon UV cleavage, when barcodes are released to the nuclei, the bead closest to each nucleus contribute the strongest signal, and the beads slightly farther away will give you the weakest signal. So this diffusion forms almost like a GPS fingerprint. So I'd say we now have an air tag for each nuclei that is going to be associated where they are, and you are going to be able to track it back where each nuclei comes from after you already have your very sensitive single cell data. And because each nucleus absorbs multiple barcodes, we basically triangulate each exact XY position, and it gives us about 3-4 micron accuracy. So how do we convert these information barcodes into precise XY coordinate? As mentioned earlier, after the UV release, spatial oligos diffuse into and tag the nearest nucleus, and each spatial oligo on the tile has an already known coordinate. And we know this because we sequence each slide, and that each slide has a barcode that you can refer to at the downstream analysis. And after the sequencing, the Tracker analysis pipeline looks at all the barcodes captured inside each nucleus, and they form a cloud of points. And the pipeline then identifies the densest region of that cloud and compares the weighted center. So it looks for the most centroid location, and that is going to give you the nucleus exact coordinate. Now, let's look at the workflow. The workflow is only in 1 hour upstream with no instrument requirement. Very simple workflow from your single cell sequencing. It starts with placing your tissue, and 25 micron, we do think that the sweet spot, which again allows you to have your intact nuclei with the 25 micron, and you can go up to 30, and depending on your density of your tissue or nuclei content. And then with the UV cleavage, now all the barcodes are absorbed by the nuclei, and then you isolate nuclei tagged with spatial barcodes. From this point on, you use your choice of single cell platform, and whether it is microwave based or droplet based, or you're interested in combinatorial indexing to do higher multiplex, you create two different libraries. One of them is the tracker library, and the other one is gene expression library. And the tracker library does not require as deep, you have to do, depending on the platform, 1,000 to 5,000 reads per nucleus. And at the end, with your FASTQ files put into primary pipeline, you will be able to put your nuclei spatial coordinates on top of your gene expression data. Well, some people ask if they can do a staining. So we can't really do immediately directly on the slide because it is, you have to be pretty quick with your nuclei yield and quality. And also it is a destructive process. At the end, you have to remove your tile and you have to extract your nuclei. But you can take an adjacent section in H&E stain. And I'm gonna use this slide to help you understand that, but also to really highlight how accurate the positioning of the nuclei is. Sorry, do you have a question?
Go back. I don't understand how the tracker probe pairs to the RNA. Does it?
It doesn't. It acts like a separate RNA. So when you isolate your nuclei, your barcode sequences act almost like other RNAs within your sample. But when you do your, let's say, 10x protocol, your library for the tracker is going to be in your supernatant, and your pellet is going to be your gene expression. So we have our barcode-specific primers that is going to also create two separate libraries. But they are going to act like any RNA sample within your sample through the
Oh, I see. So you're basically doping in a library of these tracker oligos into the different cells in there.
Yes, yes. So it is like a combination of, so that there are like millions of oligos in each barcode. And when barcodes release these oligos, and that they all have diffusion patterns, like think about that all the different colors, but depending on what oligo combination representation we have, we can track them back to the XY coordinates through the pipeline, but they are being treated as a separate library all the way until the end, where we bioinformatically put them together through the tracker pipeline.
So is the tissue you're putting on there fixed or not fixed?
So that's a great question. We started, and I'm gonna reveal all these things layer by layer, but we started with fresh frozen, not fixed, as depending on the downstream. However, don't quote me on these, but we are trying different kits and using frozen fixed nuclei to use it on the FX kit, the Flex protocol, or we have now an FFPE compatible protocol. But ideally, we work with fresh frozen tissue, not fixed cells. So the idea is you get your fresh frozen tissue, you put it in, you recleave, and quickly work on your single nuclei isolation methods that you're comfortable with, or we provide the FAS support and the protocols for it, and that your nuclei yield and the quality of your nuclei yield is ultimately your resolution. So we want the best out of it, and that's how we are going to get your spatial map.
So this melting technology doesn't...
That is, yes, I know that's deceiving, but basically when I cut a section on a cryostat, to make sure that it's fixed, like what I used to do is I put my finger and slide through it. That is what really melting is, and but it is not like literally with a heat or things. Just that in a cryostat, when you put your slide, either carry it over or I used to tab it. Melting, I mean by just kind of putting your section tightly on that slide so that it doesn't fly out or it doesn't wrinkle. Like any other spatial methods that actually also start with a visual or with the capture base, you have to start with a slide. Usually they're only 10 micron, and that is also requiring you to put a very even nice morphology section on the slide. And I know it will make more sense as we go along, and I'm happy to come back to really how we tag it, because that is ultimately the basic and foundation of it. But it is a very simple approach to, like, the way I say it is that you tag each page of a book. If every page was a transcript and this book, but I tagged the book, which is the cell, or which is the nucleus, and now I know that all the pages there already come from that book. Instead of trying to, like mRNA transcript, trying to tag all these mRNA transcripts like this, other capture method, and not trying to guess and put what pages belong to what book. So that is one other analogy I thought of to explain the difference.
I'm just struggling with the biochemistry though, because on the one hand, you're able to melt the cells cytoplasmic membrane to get this checker tile of libraries into the cells, but then at the same time, you're then able to digest the cells to get the nuclei.
Yeah, I mean, that is just regular protocol that you normally would do. Normally with the nuclear isolation, you would get a chunk of a tissue and put in the nuclear isolation steps. But in this case, we are, in a way, concentrated your sample by using the 25 micron section, and melting shouldn't really deceive you. Like, we are not really hurting the tissue. It is just you're putting the section on the slide. And again, that's how you start with any other slide base, which all spatial platforms are, and the same, technique that you would do.
How many nuclei do you tag? Do you tag all of them? But then the more important question, how many nuclei do you actually capture at the very end? Like, what is the percentage?
Yes.
Because I'm pretty sure that embryo, there's like a lot more than 50,000 nuclei.
That is 50,000, but I will again show you and answer all these questions along the way so that it makes more sense. And I have answered all of your questions. I'm prepared. And that is a great question, but whatever your we estimate, and that includes the loss through the sequencing process, the aim, like if you're working with a 10x, right? If you want to aim 10,000, they all have parameters, how many nuclei you have to start with. And the BD Rhapsody actually gives you an option to load 100,000. And then it lowers the cost, and we, because of our connection with the BD Rhapsody, we actually develop a lot of these things on BD Rhapsody. And I also have public data sets that I put a QR code for, so that you can actually look at the data sets and see it for yourself. But I agree with you, and that is the biggest concern, and I will highlight how we do that. But just to highlight how accurate that tagging is and how dense that data is, again, to remind you, this is about like 55,000 nuclei from 11-day mouse embryo, and it's exactly expected as a developmental stage, and I don't have any nuclei diffusion on the ventricles, and it is very clean. So now with this ST-Align tool, you get the best of two worlds, right? When we merge these, you get the great anatomic morphology as well as your spatial nuclei. So now I already know where all of these cells come from. And just going back, what we keep on talking about capture-based methodology and how that changes in a data level. Then this is a direct comparison on the mouse brain, and you see how the using capture-based method, the UMAP looks like a blended continuum, and that is one of the biggest challenges with the diffusion. And as each of these bins contains mixtures of multiple cells, and to recover, you need the convolution algorithms. And even then, your true cell boundaries could be ambiguous. But on the right, that I will show here, because we are relying on the single cell sequencing, you see your beautiful clusters as you would see your single cell sequencing, but because all of these nuclei we isolated have spatial identity integrated to them, we are able to bioinformatically space them back into where they should be. And without any transfer, integration, or computational stitching, the same clusters are now projected directly onto their spatial coordinates. And to answer your question, yes. So when people think about how many nuclei I isolate, I just want to start with the tissues that we are most comfortable with, but also it's easy to validate because the brain anatomy is very distinctive when you think about compared to other complex tissues. And from a 25 micron mouse tissue, we isolate about 150,000. And when we follow the general 10,000 nuclei mapping, you can actually see most anatomic structures, as I can already identify the cortex, hippocampus, and all these layers. But because the density of your data is, which is your resolution of your data, is directly linked to how many nuclei you run, when you increase that to 30,000 single cell, you can actually see the finer structures revealed, and now I can really identify different spatial layers, and then I can really identify all of the different structures. So Tracker scales beautifully, loading more nuclei gives you the denser and more resolved maps, all from the same tissue section. And now, going back to why you're concerned, because when we think about a bigger chunk tissue, you isolate, and when you look at 3 million, yes, you will get some cluster. But if you were theoretically put that in a spatial platform, yes, that wasn't going to be meaningful and your information was going to be sparse. But the important thing to remember here is we kind of, sorry, I don't know why that animated, we start already with a 25 micron. So we are already concentrating our samples. So it gives you a very dense, meaningful information that is also going to help you to answer a lot more biological questions in terms of like secondary analysis. So you can go up to 30, 50,000. We always recommend overloading, as single cell experiments usually undersamples. Then you will get this data. And again, I have a couple more real life examples that I will be happy to present. But this is something I just like showing as a side advantage. Because Tracker captures a much higher fraction of nuclei, as I mentioned, the high data concentration directly impacts what kinds of biology you can do. I don't know why I didn't get water. Here's an example from the embryonic liver. I'm gonna see if this works. And on the top is our full 30% nuclei recovered spatial density data, and how I can get the perfect cytokine interactions. Can you get me spike water? Thank you. I don't know why this happened. I'm not used to being so loud. And the signal landscape is complete. All major interactions are visible. The bottom is kind of the theoric, artificially subsampled. I couldn't be a terror artist, but it's okay. Which resembles the sampling of standard snRNA sequencing, and most interactions that you would see with that dense data disappears. So we are compatible with all these different platforms, Parse, 10x, PD, Rhapsody, but are we just satisfied with the RNA expression data, or do we want to do immunoprofiling and multi-omics like ATAC sequence? So one of the biggest advantage of Tracker is flexibility. You don't need to buy a new instrument or commit of a new assay ecosystem. It plugs directly into your workflow. So as of now, we're compatible with PD Rhapsody, Illumina Single, Parse, Scale, 10x, and for the 5' gene expression, we can also do the 10x, and we have different kits for different protocols. So we started with the Tracker U. That was the original protocol that was doing the 3' gene expression. And then Tracker Q is one of our newest edition that is using the Parse. And I will mention the quick workflow on that. And the 5' kit is also very recently launched. And the most exciting product that is being early access, and we are able to get it, is the FX kit. So I'm gonna move on.
With FX, it's gonna be with fixed cells.
Yes, so that is the biggest advantage of it, that not only you're highly complex, and for people who have logistic issue where isolating nuclei and fixing needed, so this is actually going to be a great solution, because after that you can isolate your nuclei. So it is going to give you the flexibility of timing and batching.
So you mentioned 25 micron sections. Is
Correct.
it thickness or not?
25 micron section, yes.
Thickness.
Yes. And we have gone up to 35, 40, and we have the training slides that you can optimize. And this is just like same slide, but not barcoded, or you can take a curve and do your nuclei isolation protocol just to look at your regular output count, and before you actually use up the whole barcode slice. And I know we started a little late and we're short on time, and I'm happy to continue, but I just want to make sure everybody's time commitment. A few more data slides I would like to overview. So just checking, any questions I can pause, or are you okay for me to continue? Yes, please.
Is this platform just been tested with mouse and human, or is it tested any other model organisms?
So that is another excellent, great question, which highlights. So most of these spatial platforms, and I was hoping to use that for as an example, is that species fixed, right? Because most of the probes are using human mouse thread, and because you are relying on just RNA capture and sequencing, you can use it for zebrafish or cyno and any tissue that you can isolate nuclei, and you can do single cell sequencing. You can add spatial content to, except FX, because that uses 10x flex kit, which is probe based. Unless you do custom, then you can design other species. But it is important to say, I guess, that it is instrument agnostic, downstream assay agnostic, but also species agnostic, as long as you do the first set of assays. And so this is the data that we mentioned, the brain, and I just want to highly overview it because I want to, show that not we are only doing the brain, but what I want to really show here is that, how simple, straightforward annotation without complex deconvolution or segmentation is possible, where each dot you see here represents a nucleus. And so once you get that clear, distinctive structural maps, and even though in the cluster you might see a cell group as one homogeneous cluster, with the tracker you can actually now dive into and resolve that cluster even in the spatial heterogeneity. And this is a great example where we show astrocyte 2, even though it looks like a single cluster on the UMAP, when we put it back and zoom in, you see that astrocyte 2 has very multiply spatial distinct subtypes, like L2-3, and then we did L4, L5, L6. And not only spatially they are distinctive, they also follow very specific expression signatures depending on that layer. So it gives you the ability to break open the heterogeneity inside an already defined cluster. And beyond this one population, tracker reconstructs whole anatomical structure with subtype fidelity. Like in hippocampus, I can see my CA1, 2, 3, and that gyre area. I can see the good laminar sections in the cortical layers, and all the positioning for thalamus, hypothalamus are exactly expected. But even microglia is a great example to show here because it is quite challenging self-type with other imaging or capture-based methods. But because we are tagging them and they are not disappearing within the higher expressed, bigger, more RNA content cells, and that we are able to spatially isolate them. So we have presented a lot of brain data, and I know, but I just want to show how tracker also helps you within your complex tissue where your interaction in your microenvironment is crucial. And so this is a human ER positive luminal breast tumor sample. And this is profile using 31,599, let's just say 32,000 nuclei. On the left, you see the UMAP, and there are specific clusters that I am able to identify, LUM1A and A2, and then I have my cells as expected, T cells, B cells, and so on. But when we back these nuclei back into space, something striking appears that the completely segregated tumor subtypes are visible, luminal A and lum A1 and 2. So this separation is not obvious in the cluster from the single cell sequencing alone, but with the tracker, now we reveal the underlying anatomical department and the microenvironment shaping them, which is actually hinting us maybe some different type of interactions are happening. So when we zoom in further, the separation becomes even clearer. Spatially variable genes sharply mark the boundary between LUM1 and LUM2 tumor group, and draw a clean border. And the LUM2 is specifically interesting when you look at the cell interactions, and as it shows enrichment for CD4 T cells, cycling T cells, memory B cells, suggesting a more immune active microenvironment compared to luminal 1A. When we use cell check to examine ligand receptor communication, that confirms further where we see strong signaling between the cancer-associated fibroblasts, I'll say calc from now on, and T cells and macrophages, B cells. So the tracker is not now separating tumor subtypes, it is revealing where all these cells are, what genes define these boundaries, and how microenvironments communicate. Again, the spatial insight that you would have lost through the single cell sequencing data is put back into context via tracker cell. And I mentioned multiomics. Multiomics is a very similar protocol, but you follow your ATAC or VDJ protocol. And the VDJ, you're not expected to see so much of your TCR, BCR. However, I'm happy to share our poster. And if you already have successfully managed to get if expanded clonotypes or CAR-T, we are also supporting that protocol to do VDJ. But whether you are working on ATAC or gene expression and the tracker spatial library, you're able to put that upstream to your ATAC multiomic workflow as well. So here is the multiomic data we generated from the mouse kidney, and where we generated both single nucleus RNA seq data. On the left, you see the clusters through the ATAC seq, and that you're able to identify all the chromatin accessibility. And on the right, our RNA-seq data, where we are able to define different distinctive cell groups, and each color corresponds to a known kidney cell type. And tubule segments, collecting dot, macrophages, fibroblasts, etc. And that takes all these same nuclei, places that back into the tissue, and that is what the spatial map on the right is. Now, because all these information is associated with that, and it basically, the tracker spatial data acts like the backbone of both data that we're able to look at it. And let me kind of show that in a more zoomed in way, that spatial RNA expression appears exactly expected. Genes like NAPSA, GPX3 show region-specific patterns reflecting known kidney zonation. In the regulatory layer, you can see distinctive chromatin accessibility peaks that is differentiating, distinguishing S3 versus S1. And on the right, when we combine both of that information and this expression for GPX3, you can actually see all the linked regulatory peaks that correlate with the expression. So now it gives us both modalities, spatial scaffold, and only from one tissue, one experiment. And this is another multiome data from the human melanoma sample. And again, the cluster that we identified, malignant 1 and malignant 3, follows a very distinctive pattern when you put them into spatially mapped configuration. And the tracker lets us now 3 layers simultaneously, right? We have the transcriptional phenotype, we have a regulatory program, and the exact spatial location reveals the tumor H population that is epigenetically primed for invasion. We kind of see it from this location when you look at the edge of the tumor, but also with all the regulatory DNA motifs, it is all in line with the invasive regulatory of it. And this is simply just to show you how simple the pipeline is. It is cloud-based, or you can download it to your system. And you have the 2 output that you generate from your sequencing, and tracker analysis pipelines puts them in, and you have this great output data that will give you some of the report efficiency of your nuclei positioning. That is now, you can get better information. But to go back to your question, if you follow your sequencing track, guidelines, so if you do for 10,000, if you have to do load 30,000, this should be in line. But we say, if you start 50,000, expect 50, 60,000 depending on the platform, like 100,000, I guess 50, 60,000 will be accurate. It depends on really the tissue and your nuclear isolation protocols and the skills. And finally, the report provides a glimpse of all the biology and the UMAP and what the spatial embedding looks like, features that are spatially variable. We have a great guideline if you are bioinformatics savvy and if you want to do your own secondary analysis, you can use any of these tools. But you can also just use LatchBio and BioTuring. LatchBio is, both work with all the single cell data, but they have a great database, and they also are compatible with all the tracker, seeker, and data, as well as other spatial. So it is actually giving you a great way to, oversee everything in one platform, and that way you can really look at a lot more biology in terms of, cell-cell interaction, pathway analysis, and so on. And as I mentioned, we have these public data sets available, and all you have to do is get on the website and fill in this form. So that will allow you to play with the data, and I'm happy to connect you with our field application scientists and bioinformatics that we can walk you through it. And also hopefully with the core, we will run some pilot data projects so that you can see. And before I wrap up, we are very excited, as I mentioned, for people who have clinical samples, who wants to be able to utilize FFPE samples. We have the FX kit coming up, and it is very similar workflow. The only difference is, which gives you flexibility to do the imaging now, because your tissue is on a slide, and you put the tracker tile on top of your slide, and we have an alignment tool that perfectly keeps them together. And when you recleave the tissue, the barcodes are going to be again donated up to your tissue. And now from that point on, you're going to follow the 10x FX protocol. We have a webinar tomorrow, and if you sign a register, but if you can't log on, you can always get a recording or let me know, I can share. It is going to be on glioblastoma samples, so some exciting new data. I think it will really help you to polish what I have just gone through today. And so simply, if you have not done any spatial and you want to start from somewhere, and if you're already doing single cell sequencing, Tracker will allow you with 1 hour only to get all of your spatial mapping integrated to your very sensitive data. And that way you have now the power of integrating single cell sequencing data to transcriptome or a genome of single cell all in one essence. Thank you.
Before start, could you share the slide?
For the webinar?
Yeah, I mean the whole slide. Do you mind to share the presentation?
The presentation?
Yeah.
Yes, we are going to be signing. That's why if you have not registered in the form, and if you don't have your email, I have put a notebook there. Please put your email so we can actually email you the slides.
Thank you.
Go ahead. All right.
They're shut down 20 seconds.
So I'm going to come closer. So we are going to stay behind. If anyone has time, and if anyone wants to do a little project discussion, I know we are going to meet soon. And I have also Jane, my manager here. We can both take some spatial questions. I need to figure out I know, I'm like, sure.
So I guess
We're trying to finish libraries this week. And then maybe give it to you the 15th. Do you have the kids?
So that is only the label you can the online. So those were not labeled